Bismillahirrahmanirrahim. Assalamu alaikum. My name is Usama Zafar. My registration number 18 Arid 6494. I'm student of PMAS Arid Agriculture University Rawalpindi. Today my presentation topic is clover from course breeding fodder and a forage crop. We will discuss about the introduction of the clover. Scientific name Trifolium resupinatum. Common name Persian clover or shaftal clover. Family Leguminaceae. Feed category legume forage and a forest plants. Related feed persimmon and a white clover. Origin and history of clover. Persian clover is native to Central and the Southern Europe and the Southwest Asia. In Europe, it was cultivated as an ornamental plant for many years. Now it occupies a significant position as a fodder crop in Egypt and Iran, in somehow area of Pakistan and Afghanistan. In Pakistan, Persian clover is also relatively new fodder crop. It appears to have been introduced in 1910 to 15 in Lahore region, Pakistan. Now we will discuss economic importance of a clover. It is a multi-cut crop. It can be cut 2 or 3 times, and in irrigated field optimal cutting interval were found about 6 to 9 weeks in growing season. It can be grown open range land for grazing purpose. It is used as a fodder, supplying high palatable and nutritive pasture and hay. If the fodder feed alone in excess amount, it causes indigestion in cattle, so it should be chopped and mixed with the wheat straw. It is a soil binder and a nitrogen fixing crop. It improves the soil fertility. It is a symbiotic relationship with the nitrogen fixing bacteria. It attracts aphidophagous arid that play an important role in reducing the aphid outbreak in vegetable crop, which is known as biological control of aphid. It is also used as a green manure. It improves the soil fertility. It is mostly used in Asian countries. It is a nutritious forage rich in protein and a mineral. It contains 14 to 27% crude protein and a 14% minerals. Now we will discuss about the floral structure. Inflorescence is flattened head with the highly fragrant pink to purple flower petals having a resupinate corolla, hence the name resupinatum. Single seeded pod split open at maturity. Seeds are tan to reddish brown and very small. Stems are hollow, branching from the base. Leaves are trifoliate with a 0.4 to 1.2 inches long to oval, oblong, relatively fat. Each inflorescence consists of 20 to 40 florets, which are white and commonly trimmed with pink. 3 to 4 seeds per pod can be produced after pollination. The seeds are the smooth, heart-shaped, and the range is colored from bright yellow to yellowish brown and darkened with the age. Now we will discuss pollination and the pollen dispersal. As Persian clover is predominantly self-incompatible, cross-pollination is essential for significant success. Persian clover pollen is not easily dispersed by wind, and an airborne pollen does not result in effort pollination. Therefore, insect pollinators are normally required to transfer pollen between individual clover plants like bumblebees and honeybees. Emasculation method in clover. Prior to applying the emasculation treatment, all flowers on a head except those to be treated were removed using a pair of a tweezers or a wing and a keel petal of 20 to 50 newly open flower at center of the flowering head were removed, thus exposing the stamen and the pistil. This was accomplished by the grasping an uppermost part of the wing with the tweezer or forceps and using a sudden upward jerk. That head was then immersed in a beaker containing the proper ethanol concentration for a period of 10 to 20 seconds and then rinsed in a beaker of water. The head was then either left untied, rolled with the finger, brushed with a cotton swab, or pollinated by an unrelated genotype. Concentration of 47.5, 57, 66.5, 7.60, 85.5 and the 90% ethanol were used. Immersed then the head in water alone was used as a check. When the flowers were left untouched, no seed was obtained, but the seed was obtained when the head were recalled or when the wing and the keel petal were removed. Rinse the flowers with the water. After removal of a wing and the keel petal failed to effectively control self-fertilization. The work of Atwood in 1941 in Folium resupinatum emasculation of the flowers by use of suction was not found to be effective. This is undoubtedly due to the fact that the pollen grains settle on the stigmatic surface as a result of removing the wing and the keel petal. The stick cannot removed by the suction and are undistributed by the water alone. Selfing method in clover. Selfing by rolling the head between the fingers after the ethanol treatment invariably produced no seed. Since this was thought to be due to the injury of the export, a gentler method of pollination was used. A piece of cotton was rubbed across the anther and the stigma to cause self-fertilization if any pollen was still viable. Especially no self-seed was obtained using this procedure except when the head had been soaked in 66.5 ethanol of 10 seconds. Now, crossing method of clover. In an attempt to ascertain of the effect of ethanol on the pistil, each clone was crossed to an unrelated plant. This was accomplished by removing the wing and a keel petal from the flower or on the head of the unrelated plant and gently rubbing this head over the flower already treated with an ethanol concentration. Maximum cross seed set was obtained not at the low ethanol concentration but when the flowers were soaked in 66.5% ethanol. At the first, this was thought to be due to some artificial in the technique, but when this same treatment or combination was attempted on more genotype and the flower, similar results were obtained. Self-fertilization was prevented when using ethanol concentration of 66.5 molar or greater. Although maximum crossed seed set was achieved using 66.5 ethanol, the fact that some self-seed were obtained at this concentration would suggest that most effective concentration for preventing self-fertilization would be 764. Research work on the clover. There are 7 commercial varieties of Persian clover available in Australia. There are 2 main subspecies of Trifolium resupinatum majus and resupinatum var. Trifolium resupinatum variety majus has an erect-headed, thick hollow stem and a large leaflet. Heart seed level is very low at 1 to 2%. Flowering and the maturity is mostly late. The varieties Maral, Morbal, Lesser and Lightning belong to this subspecies. Trifolium resiconatum var. Resiconatum has a more prostate habit, thinner stem and the smaller leaflet. Hard seed and the seed yield are higher than the majors. Flowering and the maturity are mostly aerial than the majors. The cultivars are Cayambaro, Nitro and a Prolific belong to this subspecies. Now seed development and the seed dispersal. Persian clover flower have been between 21 and 104 floret per flower head. Based on the number of ovules present, each floret should be able to produce 6 seeds. Seed development from pollination to full ripening takes 26 to 30 days or less than. Some seeds start becoming variable at about day 10 with approximately 90% seed very viable by the day 15. Persian clover seeds can be dispersed a long distance by the human activities, though the digestive tract of the bird, grazing animals. Alternative short distance dispersal may occur by the digestion, stocked and playing worms and to small extent by wind. Seeds can be remain viable after passing through the digestive tract of sheep, cattle, goat and several days after consumption. Conclusion about the clover from my research. Trifolium resiconatum is one of the most important vegetables of other food and a medicinal plant. The plant is rich in important flavonoids, isoflavonoids, phenolic acid, glycosides, monosaccharide, protein, essential fatty acid, tocopherol, and concentrate. Thank you very much for listening to me.